To Encyclopedia World. This is the part 2 of biotechnology. The first part discussed to you the definition and the two biotechnology in ancient and modern times. And now we proceed to the applications of biotechnology in terms in medicine and industry. As you can see in the picture, GMO are look like the original one. So you cannot define easily whether the have thing you hold is the real one or the one who composed by genetically by biotechnology. So now, like I've said a while ago, we're going to tackle about the application of biotechnology in medicine and industry. So first, we're going to start with biotechnology and medicine. Microorganisms have been used to generate variety of products useful in medicine. Some of these are being like antibiotics, vaccines, hormones like insulin, and enzymes. The purpose for biotechnology for medicine is to heal the world, which biotech is helping to heal by harnessing the nature's own toolbox and using our own genetic makeup to heal and guidelines of research by, first, we have the reducing rates of infectious disease. Second, saving millions of children's lives. Third, changing the odds of serious life-threatening conditions affecting millions around the world. Next, tailoring treatment to individuals to minimize health risk and side effects. Creating more precise tools for disease detections. Combating serious illnesses and everyday threats confronting the developing world. Biotechnology in medicine have actually a lot of processes. But in this case, we're going to discuss only the major product by use of biotechnology in medicine. The first one is the production of antibiotic or penicillin. The antibiotic penicillin is produced by microscopic fungus Penicillium, and it was discovered by Sir Alexander Fleming in 1928, and it proved to be a wonder medicine during World War II. It is still an important medicine and is used widely to treat different infections in the body. Also, different mutant forms of fungus produce different types of penicillin, which are effective in treating different diseases, like ampicillin, tetracycline, and some of the common important antibiotics. Production of antibiotic penicillin are to prevent bacterial growth by either killing the bacteria or making them inactive. The second product, we have the production of human insulin from bacteria E.coli. With modern biotechnology, it is possible to produce genetically modified bacteria that carry the desired gene and produce the desired hormone or protein. Human insulin is the first hormone produced by genetically engineered bacteria E. coli, and production of human insulin is by using recombinant DNA technology, and the gene that produces insulin is inserted into the bacteria E. coli, and genetically modified E. coli produces human insulin. And since 1982, the insulin has been produced commercially on a large scale by this method. Aside human insulin, E. coli bacteria have also been used to produce human growth factors like somatostatin and somatotropin. The genetically modified yeast cells, fungus, have been used to produce vaccines against hepatitis B virus, mumps, and rabies, and so on. So how did they make penicillin? As you can see in the picture, we have the penicillium mold naturally produced by antibiotic penicillin. And after it, we have here the deep fermentation tank, which they add a kind of sugar and other ingredient to process the penicillium. And then scientists separated the penicillin product from the molds And finally, this is it, we have the antibiotic medicine. Insulin hormone is to convert extra blood glucose into glycogen. It is produced by pancreas in the body, and in people with deficiency of insulin, extra glucose is not converted into glycogen, and so it remains in the blood and is excreted in the urine. This disease is called diabetic mellitus. People suffering from it need to take insulin every day. The second application we're going to discuss by biotechnology, we have the industry. Biotechnology in industry is the simplest, and based on biology, it is the biotechnology harnesses cellular and biomolecular processes to develop technologies and products that help improve our lives and the health of our planet. We have used the biological processes of microorganisms for more than 6,000 years to make use food products such as bread and cheese and to preserve dairy products. If the medicine is to heal the world, industry is to feed the world. Biotech improves crop insect resistance, enhance crop herbicides tolerance, and facilitate the use of more environmentally sustainable farming practices. Biotech is helping to feed the world by, first, generating higher crop yields with fewer inputs, lowering volumes of agricultural chemicals required by crops, limiting the runoff of these products into the environment. And also using biotech crops that need fewer applications of pesticides and that allows farmers to reduce tilling farmland. And also developing crops with enhanced nutrition profiles that solve vitamin and nutrient deficiencies. Producing food of allergens and toxins such as mycotoxin, and also improving food and crop oil content to help improve cardiovascular health. The first example for industry, we have the cheese. Cheese is produced by first curdling of milk by bacterial streptococcus or lactobacillus and then salting the ripening it by variety of microorganisms depending on the type of cheese to be produced. The next example, we have the vinegar. Sugar starchy material like crushed malted barley or grape juice is fermented by yeast. Yeast is a microorganism that causes fermentation and converted to alcohol. Vinegar is manufactured by fermentation process, and the alcohol is then converted to acetic acid by further fermentation by bacteria Acetobacter. The third example, we have the yogurt. The milk is first pasteurized and heated to 72 degrees Celsius and then cooled rapidly to destroy harmful bacteria. Pasteurized milk is then fermented by adding starter culture of bacteria, usually a mixture of Streptococcus, Thermophilus, and Lactobacillus bulgaricus. The bacteria convert milk sugar lactose into lactic acid that coagulate milk proteins into yogurt. The fermentation occurs best at 40 degrees Celsius up to 45 degrees Celsius. The yogurt is then cooled to 5 degrees Celsius to stop the bacterial process and otherwise it turns sour. Next example, we have the alcoholic beverages. Alcoholic beverages are prepared by fermentation of sugar by yeast and not bacteria. Yogurt and cheese are prepared due to fermentation by bacteria. The fermentation of sugar by yeast sugar is converted into carbon dioxide and ethyl alcohol, and alcoholic beverages like beer, wine, and whiskey are all prepared by distilling the fermenting product. The source of sugar differs in all. Depending on that, the product also differs. The carbon dioxide produced gives the sparkle and fizzes taste. For example, beer is from barley grains, wine is from grapes, and depend on the type of grape you're going to use, the variety of wine will be result. Industrial alcohol is from potato and molasses. The next biotechnology industry, we have the vitamins. Today, a number of vitamins like vitamin C are produced by transgenics, like vitamin B12 is produced by Aromaticaceae asvayi and certain species of Acetobacter. The next one, we have the enzymes, and two most commonly used microorganisms for commercial production of enzymes are fungi and bacteria. Genes from different organisms are introduced into these microorganisms, which then produce the desired enzymes. Applications for biotechnology summary and some commercial application uses of enzymes are given in the picture. Thank you for listening to Pedia World and hope you learned a lot about biotechnology.